Good afternoon, and thank you for joining us for the March 2023 GLASE webinar. My name is Haley Hawley. I'm the Extension Specialist with the Greenhouse Lighting and Systems Engineering Consortium, or GLASE. And before we get started with our webinar, as usual, I would like to thank our current GLASE industry and CEA members for their support. It's because of them that we can promote these activities and offer them to a wide range of audiences. I would also like to highlight one of our projects that's free to all greenhouse growers in New York State. The project is called the Greenhouse Database and Benchmark Tool, and it enables growers to receive a free energy audit and evaluation of their energy use. We already have about 10 participants. We're trying to get 80, so if we could get a few more, that would be fantastic. The results that you get can then be benchmarked against other anonymous greenhouses to compare how your energy systems compare with commercially available equipment. And you can go to the website on this slide to find out more information. So let's get started with our presentation for today. Remember that there is a Q&A box at the bottom of your Zoom screen, and you can use that to ask questions throughout the presentation. And we will save some time at the end of the presentation to go over those questions. Today, we are happy to have Dr. Yaguang Luo. Dr. Luo is a super-grade senior scientist at the U.S. Department of Agriculture Agricultural Research Service, or USDA ARS. She joined the agency in 2001 after over 5 years of research and management experience in the fresh produce industry. Her research centers on food quality, nutrition, and safety of fresh produce in the farm-to-fork supply chain. Dr. Luo has served on the Interagency Food Safety Task Force of the White House's Office of Science and Technology Policy and consulted for the United Nations Food and Agriculture Organization and World Health Organization. She currently collaborates with NASA in researching best methods for growing microgreens in space. In this webinar, Dr. Luo will share her research regarding LED light modulations for targeted improvements in product quality and nutrition traits during pre- and post-harvest operations. And with that, I'm going to turn things over to Dr. Luo.
Thanks, Haley. I'll go ahead and start my presentation. It will focus on the light modulation for targeted enhancement in produce quality, safety, and nutrition of fresh produce. Okay, here's the outline. There are 3 parts. The first one will be the LED light modulation for the quality and the nutrition of leafy greens grown indoors. The second one move up to the supply chain to the post-harvest light application for food quality and shelf life of fresh produce. And the third one will be food safety using enhanced microbial reduction by a sequential treatment of an organic acid called peroxyacetic acid and UV. Here's my first part to focus on the LED light spectrum on the metabolomic profile of ruby streak microgreens. These 2 pictures are from our CEA industry collaborators. We understand that the CEA growers are trying to use magenta light to enhance the red color and also the phytonutrient of microgreens. There are some studies, but not enough scientific information for them to make an informed decision. So we helped to fill the important data gap here. In the controlled setting in our CEA research facility, we grow microgreens under white, blue, red, and magenta light with the condition listed below here. And we noticed that the magenta light did increase the red color, especially the more like a purple color. And then with the magenta light followed by the red light also increased the red color pigment comparing to the white control. Here's the phytochemical composition. We focused on ethylphylline, polyphenols, and glucosinolates because those compounds are now antioxidants or anti-inflammatory chemical compounds or anti-cancer. So for the anthocyanin, we noticed that red and magenta LED light increased largely on the anthocyanin composition in comparison to the control. For the phenolics, we did not see a huge difference as we saw on the anthocyanin, but we did see improvement in polyphenols through the red light application. For the glucosinolate, we looked at the total glucosinolate, but also the individual glucosinolate. With magenta and the red LED light, we saw a substantial improvement in the glucosinolate concentration compared to the white light control, especially in the forms of the semi-grain composition within the glucosinolate. For the picture on the right, it's the individual component that made up the glucosinolate. So that was the first study on the general effect of different LED light wavelengths or the spectra on phytochemical composition. Now we moved on to the next project where we studied the effect of far red light alone or in combination with white light at a different intensity and schedule on the plant growth and also the flowering and the phytochemical composition. Here are our treatment conditions. We used white, which is W. F is for far red. WF is for white plus F plus far red. And the D is the dark period. And then we use the 8 hour as each segment. So some of them have WWD, which is white, dark, versus WFD, which is white plus far red, white plus far red, followed by dark, and vice versa. And then because we were interested in the color and the phytonutrient, we purposefully selected 6 vegetables from the brassica family, representing 3 red and 3 green. Here's the first picture of the ruby streak mustard at day 14. As you can see, the combination of white plus far red at the longest exposure time, which is 16 hours, followed by 8 hours of dark, had the lightest color, because the control, which is WWD, has a very intense red pigment on the cotyledon leaves versus the one with the far red, it's a lot yellower. But also we noticed that the changes in the plant maturity, as you can see, the ones with the far red has more true leaves emerged at this microgreen stage. That's for red ruby streak. Now for the golden frills mustard, we saw similar response as we saw in the ruby streak, but not as pronounced as the ruby streak. For red kale, the results are kind of opposite, because the white and the far red combination had the highest concentration. The leaves are actually more red instead of yellow or green in comparison to the white. For the green kale, we pretty much don't see any major difference at this microgreen stage. So it's really the far red treatment response is species dependent. Forward to day 27, when the ruby streak reached the flowering stage, we saw a very significant difference among the treatment. As you can see here, with the far red followed by dark, far red and the white combination followed by dark, we had a lot of flowers here. And the one with far red replacing dark, we also saw far red. But the control, which is white, and dark, we did not see anything yet, not even a floral bud. Even with the increase in the intensity of the white, no, I'm sorry, with the others, we did not see as much difference. So this is the one has the biggest difference. Okay. Now this one is the golden frills. We saw similar response, but again, not as pronounced. As you can see here, the longer treatment of white plus far red stimulated the flowers. Others are very limited, and they control, we did not see any flowering yet. Now on the red kale, none of them reached the flowering stage, and the one treated with the longest far red plus white, it's slightly larger in terms of plant canopy, but beyond that, we don't see a major difference in terms of plant maturity. Okay. And then for the green kale, we don't see any difference. None of the treatment with the far red added, even at the high intensity and longer exposure time, we still did not see anything. So because of the significant difference we saw on the ruby streak mustard in response to far red, we decided to do a focused study on ruby streak mustard. Here are the treatment condition. In this one, we added a white enhanced treatment so that it helps with the comparison with others with the far red. So in terms of PPFD and the total PFD and the DLI are relatively comparable. Okay, here are the samples on day 10. So it's still at the microgreen stage. The results are pretty much similar to the species treatment group we did earlier. However, because we added the increased intensity of the white light, we did see some changes in color and plant morphology. We saw an increase in red pigment and actually a smaller cotyledon leaf area. Here are the data as you can see the comparison. Then moving on to the baby leaf stage at day 16, the treatment difference show at day 10 continued. But again, we saw some elongation on the far red treated ones. Moving on to day 23, we started to see a big difference between the white light control and the far red added. The white light control, it is still small and immature versus the far red added for 16 hours, we saw flowers. The others with 8 hours of far red added, we did not see any flowers yet. And with the enhanced white light, which is very strong white light intensity, and then we also saw the small young plants like the control, which is white, dark. Then moving on to 27. The two treatment contains far red light started to show flowers. Keep in mind though, here in the treatment group 3, we use the far red to replace dark. So it's white, white, dark. Versus group 4, we added far red as a supplement to the white. These two behaviors are pretty similar. Keep going on to 34. The white, white, dark control still small and not mature, but the one with the increased white light intensity started to show flowers. Waited another 10 more days. Finally, we saw the flowers from the control. So in recap, the far red added, we started to see flowering at day 23, but the control did not show flower until day 44. Here's the summary. As you can see, the days to floral buds, to flowers, and to seed pod. So overall, we received the 39 to 46% reduction in terms of days to flora comparing to the white. Same to the flowers and to the seed pod. So because of this study, we now suddenly are able to get lots of beautiful edible flowers of ruby streak mustard. We checked with Google, internet, and our nutritionists. We understand that mustard seeds and seed oil are very high in health beneficial compounds. And the edible flowers from mustard are also understandably containing high phytonutrient, but we could not find any scientific data. So now that we have the flowers, we decided why not to fill the important data gap in this area. So we followed up this study with the phytochemical composition and also the entire metabolomic profile of baby green, microgreens, and flowers subjected to various treatments. So here's the first part on the glucosinolate. Sure, I read it correctly. Okay, so on the flower part, as you can see, the total glucosinolate concentration is a lot higher than the baby green. It's also higher than the microgreen. And there are treatment difference between different of the nights with or without far red and their sequence. Okay, here's the summary. Far red light effect on plant growth and flowering is species dependent. Far red light largely reduces the time between sowing and flowering. For ruby streak mustard, given the high value of the strong health benefit of mustard flowers and the seed oil, as well as the high production cost, we thought far red light could be used to significantly shorten the growth period and thus reduce production cost. The light triggering shade avoidance is known, actually it's pretty very well known. But the large difference that we observed on ruby streaks mustard is worth further investigation and certainly needs a lot more optimization. Significant difference was observed between far red light applied singularly, replacing dark, or supplementary to the white light at the equal daily light interval. So some of the findings are the confirmation of the existing science, but a lot of the new findings in this area, and we hope the information will be useful to our research partners and the industry partners. Let me check on my schedule to see. Okay, so I'm doing well. So now, moving on from the production to the post-harvest, we wanted to evaluate how would retail stores to use LED light to improve quality and nutrition. Right now, the retail stores are considering or are in the process of replacing incandescent light with LED light. Now, with the LED light, you can have all kinds of intensity and all kinds of the wavelengths. Which one is better? Lots of marketing firms and the grocery stores are evaluating the light impact on human behavior, their marketing and sales, and also the illuminance of the products, and obviously also on the energy savings part. But the research on the effect of LED light spectral on produce quality and shelf life, it's beginning, but it's a lot of study coming up, but still limited. So in this study, we compared the different of LED light spectral on the quality shelf life and also the phytochemicals. We evaluated the radish microgreens, bell peppers, broccolis, and strawberries. Here, I will show you the two studies related with peppers and microgreens. So on this slide, you can see the blue light actually caused some plant tissue injury and also resulted in decay. You can see here, this is interesting because blue light is well known for inhibiting microbial growth, especially on mold or the fungi side. So you would expect it may delay the decay, but in this case, it's more just effect on the microbial, but it's also work on the plant tissues. Once the plant tissues are injured, they could get into decay and lose of shelf life. So this one, we used the 8 degree because that's the optimum storage temperature for papers. The quality evaluation, we were able to measure firmness, color, and the visual evaluation. But we also wanted to try to see if the hyperspectral imaging could be used to evaluate the quality and the shelf life. So in this case, we used different spectral under the hyperspectral imaging, and then these are the scans of the hyperspectral imaging for different treatment of the papers. Once we are done here, we are doing additional analysis so we can get more detailed quantitative data on the comparison. I will give the broccoli and the strawberry study in the interest of time. But on the phytonutrient part, I want to share with you on one of the publications we had a few years ago. In that study, we evaluated the effect of dark versus light on the bioactive compound of radish microgreens. And we also used 2 different packaging conditions because radish microgreens can be packaged using the OTR film, which is stand for oxygen transmission rate film, which means that the oxygen transmission is selectively controlled. It could be also stored using the perforated films or the clamshells. So we want to see the effect of those packaging conditions as well. As you can see here, the phytochemical composition actually is an interaction between the packaging condition and the light-dark treatment. But in general, over time, the treatment difference changes. But within 2 days at 5 degrees, we did see an improvement in bioactive compound concentration when the radish microgreens were exposed to light. As you can see on your left, with the light exposure, we were able to significantly increase the ascorbic acid concentration, this including the total ascorbic acid, the hydroascorbic acid, and others. And then for the violnethane, we also saw an improvement with the light exposure. But again, as mentioned about, as time goes on, the treatment difference could shift. But for us, I wanted to highlight the 2 days because at the grocery stores during the display, normally you don't display there on too many days. So I thought 2 days would be a lot more relevant. And the same results as shown on the Newton, with the light increase the Newton concentration. Light exposure also increased the total phenolic compound as shown at the left bottom. For the antioxidant, we measured the different forms of antioxidant capacities, and all of them showed an improvement with the light exposure. So in this case, the 2 days of exposure to light and the treatment condition benefited the phytochemical concentration in the radish microgreens. So now what's on the quality, shelf life, and the phytonutrient part. Now moving on the last one, on the food safety part. Study was initiated to address the major food safety concerns for the fresh cut industry in terms of produce wash water, reuse, and recirculation and treatment. But with the CEA, the greenhouse, and other controlled environmental conditions, hydroponic growth conditions use a lot of, need water, obviously, even though the water usage is largely reduced when comparing to the between the open field production, first cut, and then now the CEA. So there's a huge reduction in water usage, but regardless, CEA still needs water. And water you could get from the municipal water with chlorine, or you can use other types of water which you may or may not need additional treatment. So if you do need a water treatment, then what do you use? One thing is commonly considered is UV light for water treatment. This UV light water treatment is also widely used by the treatment for the municipal water and drinking water, and also wastewater treatment. So UV is very good in terms of reducing microbial count. But as we all know, UV is also expensive, especially when you have to use a high intensity of UV for an extended period. So we were wondering, what if we do a combination of UV treatment plus organic acid mix, such as peroxyacetic acid, which is GLASE. GLASE stands for Generally Recognized as Safe by FDA. And the peroxyacetic acid is also widely used by the fresh cut industry. It's also accepted by the organic standard. So we wanted to say the combination of PAA, the peroxyacetic acid, and UV versus a sequential treatment of PAA followed by UV treatment on the microbial reduction. So here is a time curve study of the UV unknown, PAA unknown, and their combination. The increase in exposure time, as you can see here with the UV light, you have a significant improvement in terms of log reduction on E. coli population. We used the generic E. coli here as a surrogate for foodborne human pathogen, E. coli 0157H7, so that the treatment condition can be more easily manipulated. So we also did a UV intensity at 200 and 600 and 36, I'm sorry, and 360 unit. So at the PAA, we also did a different level concentration, 20 and 40 ppm. And then we did a combination of UV and the PAA. With the PAA, as you can see, also with at the same concentration, with the increase in exposure time, you do increase the microbial reduction. On the combination of PAA and the UV, you saw even more reduction on the E. coli counts. But again, as I said earlier, by increasing the UV exposure time, you come at the expense of the energy cost. So next study, we want to say, what if we separate the UV and PAA treatment? So we do a PAA pre-exposure at a various time, followed by a shorter UV treatment, which is only 30 seconds. So in this study, we evaluated the inactivation of E. coli population by the exposure of PAA at 0, 15, 30, 45, and 60 seconds, followed by UV at 30 seconds only. As you can see, there's a huge improvement in terms of the microbial reduction by doing the sequential treatment. Even at a treatment time, only half of what we used for the combined. Here is a side-by-side comparison between the UV PAA combined, we'll call it the concurrent, versus PAA pre-exposure followed by UV for 30 seconds. So for both 30 seconds exposure, the combined reached 1.7 log reduction here. We use the log here, it's the log reduction of the microbial counts over the initial microbial counts. And here, with that 30 seconds UV exposure and the 30 seconds of the PAA, our reduction reached the 3.1 log. So that was more than 1.4 log improvement. With the increased time at the 60 seconds of PAA exposure, but again, only 30 seconds PAA exposure, our log reduction reached 6.2 versus 4.4 here. So it's almost 2 log here, 1.8 to be exact. So this is really like a win-win situation where you increase the E. coli population reduction, but also reduce the UV exposure time. Again, because of that, you improve the bottom line in terms of the energy cost. Another thing I want to talk about is also the water depths of the treatment solutions. UV has many benefits, but UV also has a downside, which is the penetration to water level is very shallow. So it doesn't reach very deep. So in this case, we wanted to say, what if we pre-treat UV? We pre-treated the water with PAA, followed by combined, or followed by subsequent UV treatment. So in this chart, we have the UV alone for 30 seconds, and we have a UV and the PAA combined. And here we have a sequential treatment of PAA followed by UV. As you can see here, and then when the water depth is not high, we don't see a benefit. Actually, there was a slight reduction in this shallow water. But then when we increased the water depths, we saw a substantial improvement by doing the sequential treatment of PAA followed by UV. Oh, why do we say that? Effect here is the potential mechanism. In our earlier study in collaboration with Georgia Tech, they found out that with the PAA treatment, first, the PAA could penetrate into bacteria. You see the rod here, it's denoted the bacteria. So once PAA penetrated to the bacteria, make the bacteria a lot more susceptible to the follow-up treatment, such as UV. So that's why we saw the improvement in the treatment efficacy. So in summary, efficacy of UV disinfection of water with high organic matter is largely improved by the addition of low concentration of peroxyacetic acid. 20 ppm is really very low. The fresh cut industry currently use like 60 to 80 ppm. Sequential application of PAA and UV improve the microbial reduction efficacy and reduce the UV treatment time. Thus, potential for significant reduction in energy consumption while we still gain the good microbial reduction. One thing I want to highlight here is the water we use. It simulates the processed water with a very, very high organic load, like 1,000 ppm of chemical oxygen demand. So this is really the worst case scenario. In the hydroponic system, the water treatment, you probably don't even need this much if your organic load is lower than 1,000 ppm. So with that, I want to thank all of my collaborators and my support staff for their outstanding research effort and their intelligence and talents. But most importantly, I want to thank them for their willingness to work with our, more than just university collaborators, but especially our industry partners. This industry partner, including growers, equipment manufacturers, and also the retail and the wholesale industry collaborators. With that, I want to thank all of you for your time and attention. Thanks. I will stop sharing now.
Great, thank you so much. It's amazing how many different applications lighting can have. All right, so we have some questions from the audience that I will get started on. So first question, this is going back to the far red section of your talk. How did you combine white and far red light? For example, were separate color bulbs included in the same lighting housing, or were plants subjected to only white light for a period of time, then only far red, etc.?
Okay, sure. Actually, in this one, we have the Osram light and our study, so we could combine the different Osram light fixtures to do the treatment. However, in this case, actually, we're lucky because we were able to work with my collaborators that has 6 brand new growth chambers. The growth chamber has the function to adjust the white light and the far red light at a different concentration. So actually, it's an easy job for us.
All right, great. Does the change in flowering time upon far red light mean speeding up the grow cycle? And can that be considered a beneficial trait for microgreen growers?
Good question. I'm not sure. One thing with the microgreen is you don't want the plant to grow too tall, because I was told the consumer may not like microgreens with tall stems. However, one thing I think this is a discussion we had with our collaborator in other government entities. We were thinking microgreens, because it's small, it's harder to harvest because you always touch the growth substrate. So some others suggest, why not expose the microgreens to far red light one day before your harvest? So you grow the great microgreens, but they also help you to much easier to cut the microgreens. It's a thought, but we have never tried. If someone, the grower, wants to collaborate with us, we'll be happy to give it a try.
Nice. And good
It's a very
point.
nice thought. Yeah.
Yeah. We had a question about if there was a taste test done on these plants that were grown under far red. So we saw they had different colorations, they had some different nutrients. Did you see what the effect on taste was?
We do have a sensory specialist in our research center, and we took a lot of pictures, and we also planned to do the test for all kinds of studies, but so far we are not yet. It's in their plan.
Great. And we have one more question on the far red and the growth portion. Do you see negative effects on flowering bulbs using far red light? Does it affect the dark period of strawberries?
Affecting the dark period in strawberry. We did not do, we only did the strawberry for the post-harvest treatment, but for the growth, we did not do the strawberry study.
Yeah, have you done anything on flowering plants or on fruit-bearing plants though in far red? Do you know if that has a different effect than the greens you were studying?
We haven't tried anything else beyond the far red, beyond the ruby streak mustard. Certainly, now that we know this effect on the flowering triggering, we want to do more. But we were also kind of puzzled by such a large species dependent response. We know there would be species variation, but the large difference we saw is kind of really puzzling. So we certainly want to do more. And I thought this effect could be beneficial to the edible flowers industry, but also for the ornamental industry.
Okay. Another question just came up. I know you mentioned that far red definitely has species specific impacts. But the question is, do you anticipate that the spectral impacts you saw on these Brassica family photomorphological impacts are appropriate to apply across crop species, or will the species need their own action spectra to elicit specific compound and nutrient development?
That's a long question. Can I see it? Can I read it here in the chat?
Do you have access to the Q&A? I think it's basically just saying, do the effects that you're seeing with the brassicas that you studied, can that be put across other species, or would you need to do specific studies on other species to see what effect it has?
General principle is there, But I also expect the different response, especially if it's related with like short day plants versus long day versus day neutral. I would expect different of response, but certainly worth a lot more study, especially on the far red light, traditionally considered far red was not photosynthetically active. Nowadays, a few years ago, there was a study showing no, that was not true. Far red does have lots of impact, but in our study, it's also interesting. Most of the reports showing that the far red supplement to white light effect on plant growth. In our case, some of the treatment, we apply the far red light by itself. We also saw the difference.
Great. Do you have plans to study genetics behind triggering these genes that are related to fast growth under far red light?
Not myself, but I have a collaborator in our research center who is a great plant physiologist or more of the molecular biologist. So we talked to him about to see if there's any gene upregulation involved. Yes,
awesome.
It's in the pipeline.
Yeah. Okay, we have quite a few questions about your post-harvest and food safety section as well. So I'm going to go ahead and switch over to that. First question, it's great to see that PAA and UV treatments decrease E. coli load. Do these treatments have any effect on the product shelf life?
No. No, I should say that no negative effect on product shelf life. It's compared to chlorine, it's a little more expensive, but it doesn't have the residual like the THM, the other side byproducts residual from chlorine, but it's a little more expensive.
Mm-hmm.
Efficacy-wise, it's very good. So I myself came from the industry. So I was in the fresh cutting industry with two companies, Dow Fresh Vegetables and Fresh Express. So I know we use the PAA for higher end of the products, which we could afford.
Great. What was the turbidity of the simulated lettuce wash water in that PAA UV evaluation?
That's a very good question. I don't believe we specifically measured the turbidity of the wash water in this study, but in my other studies, we do have the correlation between the turbidity and the COD. So if you are interested in getting more information, feel free to send me an email and I will forward you other articles so that you can find the correlation.
Great. Can you speculate if pre-treatment with H2O2 could be similar to PAA or not? In Europe, we are limited to use PAA for organic produce.
Possibly, because if the function of PAA is penetrate into bacteria and then make them more susceptible, I would expect hydrogen peroxide could do something, but it's only a speculation. Certainly needs a lot more research studies.
Okay. Are there other chemical treatments that might be equal to or better than PAA? So similar question, but hydrogen peroxide was an idea. Do you think there are any others that could potentially work?
It's possible. One of my collaborators at USDA, we also conducted research with our partner in Israel. In that one, we used a combination of PAA, hydrogen peroxide, and another phenolic compound. I forgot the name. I'm not sure if my colleague Bing or are here, maybe they can add. Otherwise, that publication is out there. If you need a paper, I will be happy to send it to you. With that one, we call it the triple formula. We actually showed a very, very high log reduction in terms of foodborne human pathogens. So we could potentially give it a try. It's a special phenolic compound.
Sounds great. Lost my spot. Here we go. Have you tried sodium hypochlorite and UV application to see the efficiency of killing bacteria?
Sodium hypochlorite 15 years ago. But then I was told that the company was purchased by another company. After that company purchased, they killed the product. I don't know why, but when I used the sodium, make sure I say it correctly. Hold on. I just want to make sure I read it correctly. Can you repeat that question?
Yes. Have you tried sodium hypochlorite and UV application to see the efficiency of killing bacteria?
Yeah, sorry, I take back. It is sodium hypochlorite, so that's the chlorine. No, but we certainly could give it a try.
Awesome. We have a question from someone in Mexico. Do you think we can apply PPA UV concept for transportation of plants overseas? There are some issues of contamination.
Can you repeat?
Do you think we could apply the PPA UV concept for transportation of plants overseas?
Why? So
maybe just for
I'm
longer
sure.
post-harvest times.
It's on the water treatment. So I honestly don't know how would you apply the PAA and the UV on the transplant. I don't know. Maybe you can send me an email with more details, then give me some time to think about it with my team. I will be able to answer your question better. For now, I really don't know.
Great. And we do have a question about your email. So that we have several people who would like to follow up with you. I can put that email in when I send out the recording, or if you have a way of getting it out now, you can do that. But just a note, people are definitely interested in asking more questions and following up. I have a question just going back to far red as an overall concept. So in greenhouses that have sun exposure, how much of a difference does far red and supplemental lighting have on plants? Is that something that's much more applicable to, say, an indoor farm or a growth chamber, or does it really still have enough of an effect if you have sun exposure?
Far red normally is more concentrated in the later afternoon.
Okay. Great. Well, that's all of the questions we have. That was a really awesome discussion. We had a lot of questions. So thank you so much again, Dr. Yaguang Luo, for that amazing presentation. And thank you everyone for joining, and we'll have the recording of this ready tomorrow.
Thank you so much.
Have a great day.
You too. Bye.